Welcome to the third screencast on genetic engineering. This screencast looks at the two case studies you have to know for this particular A-level. One is the genetically modification of bacteria to produce human insulin, and the other one is the genetic modification of plant cells, rice plant cells, to produce rice plants which have high levels of vitamin A. When dealing with both of these case studies or examples, it might be worth thinking of producing a formulaic process by which genetic modification of an organism occurs, and this is what I've come up with. The first thing one does is isolate the gene that you want to engineer into your host organism. You then have to insert that into a vector. This could be a plasmid, a virus. In the case of both of our examples, it's going to be a plasmid. Transformation, which is getting our vector into our host cell. Identification is identifying the cells that have taken up the vector, which contains our target gene, and then either growth or cloning of our transformed cells. Okay, so whether it's transforming bacteria to produce insulin or transforming rice plants to produce vitamin A, this sequence of processes occur, and it builds on the various techniques that I've talked about and introduced you to in the previous two screencasts. So I do quickly go over some of those in these case studies, but you need to go back to those other screencasts and know about the various techniques in a little bit more detail from previous screencasts. So let's deal with the first example, which is the genetic engineering of bacteria to produce human insulin. Okay, I remember teaching this to some of you at GCSE. We go into this in a little bit more detail. Okay, so the first thing is isolation. Isolating the gene, in this case the gene for producing human insulin. And the way that we do this is by using an enzyme called reverse transcriptase. Okay, so what we do is we isolate messenger RNA, and the best place to find messenger RNA for producing insulin would be in a beta islet of Langerhans cell, and it's quite easy to be able to get hold of the messenger RNA because it tends to be attached to ribosomes, so that we can cell fractionation and isolating the ribosomes, we can quite easily get hold of the messenger RNA molecule. And what we need to do is to add a primer to that messenger RNA molecule, and then this enzyme, very useful enzyme, reverse transcriptase, which we talked about before, which makes a copy of the DNA. Notice that this DNA is only single-stranded, so we then have to add another primer, and then DNA polymerase to make our double-stranded molecule of DNA. Going back to the slide, it summarises all that. You've got a hard copy of this slide. So the mRNA, for instance, isolated from pancreatic tissue, B cells, messenger RNA treated with reverse transcriptase, which forms single-stranded DNA, and then that single-stranded DNA is then treated with DNA polymerase, which then forms double-stranded DNA. Okay, so the next bit is this idea of insertion. Okay, so once we've isolated the gene, next job is to insert it into the vector, and so here's typically a vector, and as we talked about before, these vectors, these plasmids, a loop of DNA, are often already engineered with genes for antibiotic resistance. Okay, in this case we've got this ampicillin resistance and tetracycline resistance. Okay, so what we do is we treat this plasmid with a specific restriction enzyme which breaks the plasmid open in the middle of our tetracycline gene. Okay. Then what we do is that we introduce our gene, which has also been treated with the same restriction enzyme. Okay. The same restriction enzyme would be used to treat the ends of our gene for insulin production, and then that would allow the complementary sticky ends to insert our gene into the middle of the tetracycline gene there. Okay, and then what happens is that we have to, having inserted our gene into our plasmid, the next process, okay, so that's insertion has been done. Okay, the next process is transformation, when we have to get our plasmid into the host organism. Okay, and the way that this can be done, there's a number of ways that we looked at that this can be done. In this particular example, what tends to happen is that what we tend to do is to use heat shock treatment. Okay, so what we do is we mix our bacteria with our plasmid. We use calcium chloride is used to help, and then we cool the mixture down to 0 degrees centigrade and then rapidly heat it up to 40 degrees centigrade, and that increases the uptake of the plasmid into our bacterium. Okay, so this is, as I've said, it can be a fairly hit and miss process for this. So the next thing to do is to identify which bacteria have actually taken up our plasmid. And again, we've covered this in previous slides, but quickly to go over it again, what we do is that we use a mixture of antibiotics and this process called replica plating to identify which bacteria have taken up our plasmids, so here are the plasmids that we want, okay, because the plasmid has our gene for making insulin in it, okay, as opposed to this and this, which doesn't. So replica plating, what we do is we plate all our bacteria out onto an agar plate. Okay, so colonies of bacteria grow. We then take a block with a velvet cloth on it, and we make an imprint of that onto a new plate which has got ampicillin on it, and that will kill all the bacteria, okay, apart from the bacteria which have got plasmid A, okay, because that's still got ampicillin gene on it, and also B, which is our target one. Okay, what we then do is that we replica plate that onto another agar plate which has got tetracycline antibiotic in it, and that will kill off our target bacteria. Okay, so what we know therefore is that these bacteria on the second plate are the ones that we actually want. Okay, so that's how we identify the bacteria that have taken up the plasmid with the target gene for making insulin in it. Okay, so the next thing we need to do is, having identified our transgenic bacteria, we need to grow those bacteria, which we very simply, in this case, what we would do is that here we go, what we do is we put them into a fermenter, and the biotechnology module would lead us to understand that we need to have this in aseptic conditions and have all the right nutrients, and to provide these bacteria with the right amino acids. And if we do that, what will happen is that these bacteria here will start to produce insulin, which we can then isolate from our fermenter. Okay, so that's the case study of making insulin. Okay, moving on to the second slide. This slide deals with the genetic modification of rice plants to produce vitamin A. Now, in certain parts of the world, there is quite a problem with night blindness due to the population having low levels of vitamin A in their diet, which causes certain problems in certain developing countries. So if we could make a genetically modified rice plant which had high levels of vitamin A, you would supply your population with enough vitamin A that this type of blindness wouldn't be an issue in your population. So what they've done, just giving you a bit of background, they've involved two genes. One gene which has to be isolated from the daffodil plant, or it can be isolated from maize, and another gene which can be isolated from a soil bacterium. And both of these genes together in a cell will set up a metabolic pathway which results in the production of vitamin A. Okay. So let's go through the whole process of how this actually happens, what we actually do in terms of genetic engineering. First of all, dealing with the isolation. Okay, so we have to isolate our two genes. Okay, so the gene from the daffodil, the gene from the soil bacterium have to be isolated from the respective genomes of those two organisms. Okay, and so the isolation process involves, these are the techniques which you learned in the previous screencasts. So these two genes, PSY and CTRL, will be isolated, this from daffodil or maize and this from soil bacterium, using restriction enzymes. So we would cut up the genomes of those organisms into different size fragments, which we then separate by electrophoresis, and then we'd locate the genes that we're looking by using gene probes. Okay, so that's how we would go about isolating the genes we're looking for. We would then have to insert these, so insertion is the next process that involves, we would insert these genes into a plasmid. Okay, this plasmid comes from a particular type of bacterium called an agrobacterium. Okay, these agrobacterium are very special in that they're plant pathogens, and they work by inserting DNA into plant tissue. Okay, so we use this as a valuable tool to get our genes into the target plant tissue, in this case a rice plant. Okay, we'll come back to that in a second. Okay, so first thing we have to do is get a plasmid out of one of these agrobacterium. We then engineer that agrobacterium so it's got recombinant DNA, where we would put in our two target genes, and also genes for antibiotic marker genes, so genes which were conferred antibiotic resistance. We would put all of those into our plasmid. What we then do is the plasmid would then have to be taken up, first of all, by these special bacteria, this agrobacterium, which is, as I say, a particular type of bacterium which acts as a plant pathogen. Okay, and that can be done by electroporation or heat shock treatment, which we have talked about in previous slides. Okay, and then what would happen? So that would be the if you like, the plasmid, transforms the agrobacterium, or the agrobacterium is transformed, taken up the plasmid. But in actual fact, the target cell is not the agrobacterium at all, but it's the plant cell. So what happens next is that the agrobacterium binds to plant cells and inserts the plasmid into the plant cell. And I've got a little animation which shows that happening.
This video is an illustration of the mechanism of agrobacterium using the genes for herbicide and antibiotic resistance. The genes are first inserted into the TI plasmid between the left and right borders. Since agrobacterium-mediated transformation is a binary system, the TI plasmid must then be inserted into agrobacterium using electroporation, sonication, heat shock, or some other method. Next, explants must be inoculated with agrobacterium. In this case, a scalpel is used to pick a colony from the plate and then wound the leaf of a plant. Agrobacterium then attaches to the plant's cell wall and transfers the tDNA portion of the TI plasmid to the host. This tDNA portion, which contained the genes encoding herbicide and antibiotic resistance, are then randomly inserted into the host's genome. The explants can be further cultured under sterile conditions on agar plates to form a callus, which is an undifferentiated mass of cells. The transformed callus can subsequently be cultured on agar plates containing the appropriate plant hormones and antibiotic to select only those plants that have been transformed. The result are whole transgenic plants exhibiting the trait of interest, in this case, herbicide resistance.
So in that example, it wasn't an example involving these vitamin A genes. It was one for herbicide, but the principles are the same, by which that bacterium inserts its plasmid, which contains the recombinant DNA of the antibiotic resistance, the CTRL gene and the PSY gene, into our rice plant. Okay, so the next thing we've done once the rice plant has been transformed, so in this particular case we've had two transformations, one transformation of the agrobacterium, which is the vector, and one transformation of the rice plant, which is actually the rice plant cell, which is the target cell. What we then have to do is that we have to, going back to here, the next process is the growth or cloning. Okay, and in this particular case what would happen is that we would sorry, I've missed out a section. Going back to that, the first thing we would work out what transformed the cells which were transformed, which we could do by treatment with antibiotics. So we kill all the cells which hadn't been transformed with antibiotics, so we were left with only transformed cells. Okay, and we'd take these transformed cells and we could grow these by micropropagation. Okay, again, this is a process that you studied when we'll do the biotechnology unit. Okay, and again we'd have studied that a little bit at GCSE. Okay, so we take our cells, our callus of cells, and we put them in different types of growth medium which promote root growth and shoot growth and so on, and we get small rice plants. Okay, and the idea is that these rice plants would produce high levels of vitamin A. So that's the second case study done. So just going back to your original revision slides that you've got a copy of in your notes. So the first bit is to do with the isolation of the gene. Okay, we talked about how we've done that. Okay, we then do the insertion into the vector. Okay, with using restriction enzymes and ligase to get our target genes and our marker genes inserted into our plasmid. We then have to transform. We have to transform two organisms. First of all, transform the agrobacterium. Okay, which is a tumeficans. I'm not quite sure how you pronounce that, but they're all part of the genus Agrobacterium, which are plant pathogens. So we have to transform the agrobacterium first, and then the agrobacterium transforms the rice plant cells. Okay, and we then have to identify. We can do that using antibiotics. Okay, to kill off plant cells that haven't taken up our plasmids. Okay, and then what we then do is we then have to grow our rice plants by micropropagation, so that we have whole rice plants, all of the cells of which have the genes for these two genes here, which are responsible for producing vitamin A in plants.